Hello folks, this is Steve AB4EL. Nothing in this episode is a promotion, nor is it a recommendation. One of the FBI's most wanted hackers is trolling the U.S. government. Apparently they have a wanted poster out for him, and he's selling sweatshirts with the wanted poster on it. But that's boring stuff. I was looking at the BBC and I saw something far more educational. I need to learn to type. There we go, BBC News. So I was looking at this stuff. And scrolling down. You know, when I was overseas, like in Israel, I used to listen to Voice of America relay station on Rhodes, the island of Rhodes. That's a Greek island. And I used to listen to the BBC. Sometimes I was listening to it directly from London, but I suspect the other relay station was on Cyprus, because at that time the UK had a large military base on the island of Cyprus. You know where Cyprus is, right? I don't need to tell you. You know where Israel is, right? So, you know, in Israel, I mainly worked citrus, but I've told you many times, I also worked in a banana plantation. Big green leaves. I get down the road down here to go to Cedar Falls, and it started with one person, but there are several people that are growing cold-tolerant banana plants. Some of them look pretty good. So I see this one here, the turbocharged plants that could boost farm output. Now this is the BBC, London. Scientists are hoping that by tweaking plant genes, they can improve photosynthesis. Folks, plant breeding has been going on for 10,000 years. Let's see if this is different. Now, first I want to see Emma Woolacott, Technology of Business Reporter. Technology of Business Reporter. Here she is. I guess she's also a senior contributor. Oh, I haven't done this before. I've been writing about technology for most of my adult life, focusing mainly on legal and regulatory issues. I write for a wide range of publications. Credits include The Times. I once got a ride in Western UK, near Hertfordshire, by a reporter who worked for the London Times. Daily Telegraph and Financial Times newspapers, as well as, but it says cybersecurity. What's the chance that there's another person with that name? Cyber News, there she is, Paydesk, New Statesman, Twitter, Facebook, Stanford Social Innovation Review. Oh, there you go. That'll fit in. Stanford Innovation Review, data mining for the landmines. AI is helping to locate landmines to speed their removal. Now that's the only article they showed. Let's see when it was published. I'm off topic already. To read this article and start a full year unlimited online access, subscribe now. Fall of 2023. Oh, now wait a minute. See, I'm interested. Thoughts in gear. I'm already off the topic. I clicked on the link. Go to the home page. This could be dangerous. Go doing well sustainably. That has to do with the plant breeding. Clients as to global news. What I'm looking for is if they'll show us the article that they can't find. All right. See, I'm just trying to track down this data mining for landmines. Comment. Hang on. Comment. Fall of 2023. It looks like it's a link. Stanford Social Innovation, Fall of 23, Volume 21, Number 4. Sign in or subscribe to view the digital edition and download the PDF. Reimagining our high-tech world, cultures of curing, how real estate development, securing trust in people and place. These are articles, presumably. But then it says, what's next? What's next? Okay. So it hasn't been published yet. It's got a lock on it. So I clicked on that link again, and it brings me back to this. So I can't read it. Now, why am I interested? Well, we're trying to see what she writes about. What is her expertise? I'm not picking on her. This is here I'll be covering in Forbes ways the content is controlled on the internet, from censorship to online piracy and copyright. You can follow my posts by clicking the follow button under my name. TikTok hit with fine. Come clean about data breaches to get lower. Dutch group major private lawsuit against. China targets U.S. voters with OpenAI new lawsuit. We're just seeing what she writes about. Business warned over risks about chatbot. X-list ban on political. So these are not very old. It's like August 23rd. So she's very prolific. You think she uses AI to help her write the articles? These are hers. Like ChatGPT has liberal bias, says researchers. Wait a minute, says the page is not working. Contact the site owner. Reload. Let's try OpenAI hit with lawsuit. This, I was actually going to do something else. I'm just trying to track down why would I be interested. You know, like when I have that playlist about Israeli cybersecurity companies, look at who the people are, their background, stuff like that. I'm always interested in who's talking to you. This is interesting. One place wouldn't let me read the article because I haven't paid. This one just says 413 is a very specific, hang on, HTTP 413. Content too large. Response status code indicates the request entity is larger than limits defined by the server. The server might close the connection or return a retry after header field. Huh? Now, that's kind of interesting. This is from Mozilla.org. I knew it was a specific one, but I tried to click on an article and the server refused to send it to me because it's too large. You know, it's all very complex and people make mistakes. I don't mean the computer made a mistake. LinkedIn. Okay. She's in the greater Oxford area, that's England. Writing and editing, University of York, so she's from Yorkshire. I remember a friend of mine in Israel in 67, 68, yeah, and his Yorkshire accent. I'm an experienced writer in tech, business, and sustainability, freelance since 1992. She's independent. I've written for most British broadsheet national newspapers, like the Times, The Guardian, as well as many leading magazine websites. Regular commitments currently include technology and other news for Private Eye, a weekly tech piece for Forbes, and that fits with what we saw in Forbes, except they won't show us the articles, and it's a lame HTTP error code. Business tech sustainability. See, the sustainability has to do with the plants. Features for Raconteur. Business tech news and features for the BBC. That's where I spotted her. And tech news for the Daily Swig and Cyber News. Never heard of the Daily Swig. Now this is the way my mind works. The Daily Swig, oh no, wait a minute. I think I came across this past week. Cybersecurity News and Views. And I thought I need to look at this site more often because the articles looked interesting. Bug Bounty Radar. Indian government flaws allowed creation of counterfeit driving licenses. Deserialized Web Security Roundup. I won't explain that. Chromium bug allowed same site cookie bypass. Ooh, let me just do that. This sounds interesting. I may find new things to talk about. Now, the thing is, she writes for them, but how can I find what articles she's put here? Maybe I could look at it. So if I do if I go Emma Woolacott, and Emma, excuse me if I don't pronounce that the Yorkshire way. Daily Swig. Okay, here we go. NIST plots biggest ever reform of cybersecurity framework, 23rd of February 2023. So that's the last article here by her. How to become a penetration tester, part 2 and part 1. Critical authentication bug in Fortinet. Boy, that was a big one. Let's try this. Daily Swig, Emma Woolacott, 29 November 2022. Fancy a career, you can tell she's English. Fancy a career in what one practitioner described as the best job in the world. Since you're reading the Daily Swig, you're probably already aware that a penetration tester, isn't somebody that reviews writing implements. In fact, of course, a pen or penetration, see, I knew, tester fulfills a specialist role that involves simulating cyber attacks on computer systems, networks, and infrastructure in order to identify a report. See, that could have been written by a chatbot. Then she quotes somebody. And she's quoting somebody, head of hackers at Bug Bounty and Pemberton. It looks like a French name. LinkedIn. Got to do a security check. Protecting your account. We got to do a puzzle. Why would I have to do a puzzle? See, this is the way I let my mind wander. Let me try the incognito window. No, they aren't going to let me in. One more. Hang on, one more. Let's go to Brave browser. Oh, I was mistaken. For whatever reason, you can't see it, but he's in Belgium. Site can't be reached. We were just talking about penetration testing. The first response from that site was to act like, what the hell do you want? What? We don't have anything like that. Let me interpret what just happened. They've been having a lot of trouble lately, especially with screen scrapers, bots, and they are overwhelmed at the moment. So you remember that 413 error message that basically said, what you're asking for is, at the moment is too big for me to send? That could be a site that's overwhelmed. It could be behind the scenes. We don't have any available CPU power at the moment to answer your request. And LinkedIn might be having a trouble like that. Remember, LinkedIn is Microsoft, but that doesn't mean anything. Microsoft made the news today with another big stupidity about cyber insecurity on their part. And it wasn't somebody renting space, it was Microsoft itself. And then, when the machine recovered, it jumped into the let's test and see who you are mode. Now, I'm wandering around, but she claims to be writing about things like cybersecurity, but sustainability. Is there a link between them, or are those just her interests? Now, I'm not picking on her, and this guy has a Twitter account, and by the way, the LinkedIn was be.linkedin. And that means he's in Belgium. So this is the name he goes by, and it's in this town or city in Belgium. It's right there. So let's go look and see where he is. Oh, there's the town square. What does that tell you? What does that first picture tell you about Belgium? It's been there for 2,000 years. The Romans conquered it. You can tell by the style of facades of the buildings. So let's see where, just for the fun of it, let's see how close it is to France. There's Brussels. I have a buddy there's gone. So it's kind of right in the middle. This is France. This is the Netherlands. This is Germany. And he's right in the middle with a French descendant name, at least family name. The personal name I don't know if he joined in 2012. I know which one they're talking about here. The help desk let the hacker in. Ticket trick. How I hacked hundreds of companies through their help desk. What video did I just put up a short while ago? Caesars and the MGM casinos are still down. Yeah, and how'd they get in there? By tricking the help desk. See it? See how I link all these different things that you think are totally unrelated? It's all a spider's web. So let's get back to Emma. Not the Daily Swig. Let's get back to, not there. We saw her LinkedIn. Let's get back to this. The agricultural article. Sustainability. We investigated the writer. Let's investigate whomever this is about. Improving plant yields is one way to cut the shortfall. It's called plant breeding. And there have been great advances. But while maize yields, for example, have tripled over the past 100 years, so has water usage. We need to be able to increase productivity without increasing further demand, particularly in terms of water, says Professor Steve Long of the University of Illinois. Now, they started off by mentioning maize. M-A-I-Z-E, stiff, stalk, synthetic. The plant genome, genomic variation within the maize stiff stalk heterotic germplasm pool. July 2021. The stiff stock heterotrophic group in maize is an important source of, now before I read further, they're going to say B73, B37. Is an important source of inbreds used in U.S. commercial hybrid production. Founder inbreds B14, B37, B73, and here's one I have never heard of because I've not been in plant breeding for a long time, 30 years. And to a lesser extent, B84 are found in the pedigrees of a majority of seed. Now listen to what it says. In the pedigrees of a majority of commercial. Now, this stiff stock synthetic germplasm pool, what was the favorite cross of B73 that made the corn belt explode and caused the person to write about maize the way they did? I'll say B73 XMO17, Missouri 17. Now, the National Library of Medicine cites all kinds of scientific stuff because you never know, a doctor or health researcher may find something. Expanding the genetic map of maize with the intermating B73 by Mo17 population. I'm looking and I know this name right here. Alan Allar, I've met him. The effects of intermating on recombination and the development of linkage maps were assessed in maize. Progeny derived from a common population. They went out to a cornfield. B73 by Mo17. Before and after 5 generations of intermating, they took that hybrid and then they took the seed from it and they grew those and they just let it open pollinate. Now the way maize works is heterosis is you have population A that has no relationship genetically with population B. And you try to find inbreds in each of those that they look like runts in the inbred seeds, but when one pollinates the other and produces seeds, and when you grow the hybrid seeds, the F1 generation, the plants are giants. By giants, I mean their ears are incredible compared to what their parents look like. You've seen that in humans occasionally. Intermating resulted in nearly 4-fold increase in the genetic So why did I take this excursion off to the side? To show you why I'm curious about what this guy's doing. Because he's not doing it by traditional plant breeding. So how is he doing it? If he were doing it by traditional plant breeding, they wouldn't have written an article about him. I can already guess that. One aspect of plant growth that hasn't seen significant improvements is conversion efficiency. How effectively a plant converts solar radiation to biomass through photosynthesis. Why are leaves green? Professor Long says that photosynthesis in current varieties of crops, like wheat and soybeans, has barely improved in decades. He is the principal investigator and director of a project called Realizing Increased Photosynthetic Efficiency, RIPE, which aims to genetically tweak plants to increase their yields by improving their ability to photosynthesize. And there he is, and we can see he's not 23 years old. And he's playing with soybeans. And what's the crop in the background? Even though it's blurred, you should be able to know immediately. It's maize, corn. Have to be careful about saying corn because that confuses people from Yorkshire. When you say corn, they think you mean barley. Let's just search on that for a moment. It has a web page. Ripe. Okay. Now, the team. We're interested in the team, just like I do with the Israelis in the cybersecurity. Let's find out who they are. What's USDA, United States Department of Agriculture? What's ARS, Agricultural Research Service? Lisa Ainsworth, Deputy Director. Doug Allen, Research Scientist. Mitchell Altschuler, Intellectual Property Officer, University of Illinois, Urbana-Champaign. He's the patent guy. Ali Arp, Communications Manager. Let's get down to the technicians. Javier Altaid, I can't pronounce his name right. We're going to search and see who these people are. She's a graduate student. That means she's doing at least a master's, but probably a PhD. Graduate student, not postdoc. Oh, yeah, there we go. So, Juan Guril Bic, postdoctoral researcher. See? Jose Barrero, research scientist, Commonwealth Scientific and India CSIRO. Where's CSIRO? Is that Australia? Come on, come on. Yeah. The Commonwealth Scientific Industrial Research Organization, CSIRO, is an Australian government agency responsible for scientific research, among other things ag. Have I ever known someone in the States from CSIRO? Yes, but it was a long time ago. And so my mind is rusty. I was unsure. So why is Jose, so do you think he's an immigrant there? To Aussie land? Oz? Carl Benacci, USDA ARS. Here's another one, Ryan Boyd, postdoctoral researcher. Here's the technician, field and greenhouse technician. Joshua Capili, she's a professor. Carmo Silva, Lancaster University. Do you think she's from the UK? Or do you think Lancaster University's in... Why are they doing that? That's not what I'm highlighting. Oh, I see what it is. They're grabbing the one above. Let me... Hang on. I'm curious. I think she's Italian. Lancaster University, Rubisco. What's Rubisco? That's the main set photosynthetic, photosystem 1, polypeptide. Oh, there she is. She's in a YouTube video, so there's probably, but Lancaster where? Is this international? She received her bachelor's degree in plant biology at the University of Lisbon. I could tell by the way that Elizabeth was spelled that she wasn't American born, but she's very international. Specializes in regulation of rubisco by its molecular, and she's been at ROM. Yeah, this is in the UK. I'm good at guessing. Yeah, there it is. Lancaster University, UK. There we go. Graduate School of the Environment. And it's academicuniverse.uk. There we go. So I'm good. And oh, University of Essex. That's so easy. But she really is English. Kavanaugh. Cindy Chan, data manager. She does the computers. That doesn't surprise me. Antonio is University. Antonio, where do you think he's from? My guess is Mexico. I do the same thing with the Israelis, right? I tell you which one are Sfaradim, which one are Ashkenazim, which one are Mizrahim. It's interesting to see what kind of family names they have. So there's a big, so we keep mentioning the UK, and they've got a big team. Oh, wait a minute, Urbana is known for it. Ag research, experiment manager, finance director, senior scientist, Gonzalo Estavillo, Commonwealth Science. He's from Australia. And I can tell by his face that he was born in Australia. Peng Fu, postdoctoral researcher. Benjamin Haas, crop transformation pipeline manager. That's an interesting title. They got a lot of postdocs there. Look at that. Postdocs. This guy is honorary. What does that mean? Honorary fellow. It means he's past retirement. And they're paying for him to come to the States because he's got a lot of this. He's a professor. This is really a great team. This guy, Armin Kalikian. Do you think he's got an Armenian background there? Oh, here we go. Tracy Kohler, molecular genotyping. And they misspelled the word tobacco. They have it CCC instead of CCO. Genotyping and tobacco transformation. So what they do is they pull out segments, and they put them into a certain tumor virus, and they infect the culture cells of tobacco. Oh, I knew somebody who invented that all the way back in the late 70s and early 80s. Late 70s. Amit Kumar. Boy, what a team. This is really something. This guy has got a University of Cambridge, but with that name, I'm almost sure he's Dutch. Shall we try to see if we can figure out where he's from? Wait a minute. He was a university lecturer, University of Cambridge. He's been crossing it with a maize genome and he got his degree at the Wagner. Hang on. This is the way I investigate, folks. Whoop, up. Cancel. Wait a minute. I'm guessing that he's Dutch or Belgium, but Dutch. Wait a minute. What he got his bachelor's and master's in this place. I'm not recognizing the name, and it's in the Netherlands. Ah! Because the URL ends in .nl, Netherlands. I once had a housemate from Holland. He went on to be a department head and an important international researcher. Yep, I'm good at guessing this stuff, don't you think? I love doing it. It's just like I do to the Israelis. Now, back to this. Let's find out about the head guy, because we didn't see his name up front, did we? Let's see if we can find his lab page. University, there he is. Academic appointments. E. Ken Berry, Endowed University Chair of Crop Sciences. He's got a long research history and they value him. He has an endowed chair. That means there's money in the bank, in stocks and bonds and whatever, earning interest, and it's enough money to pay his salary, irrespective of what the Illinois legislature does with the university budget. Fellow of the Royal Society, Center for Advanced Studies. He's a professor. He's on the Carl Weiss Institute for Genomic Biology, University of Illinois. See, there's these institutions named for somebody who generously funded it. Remember, we're going to something different. The Carl R. W.O.E. Weiss Institute for Genomic Biology is where science meets society, an interdisciplinary institute dedicated to transformative research and technology in life sciences using team-based strategies to tackle grand social challenges. So do you think plant breeding could be under that umbrella? I do. And faculty fellow, now this fits in with what they're doing. National Center for Supercomputing Applications, University of Illinois. Supercomputing goes back to 2001. Now, I remember when a friend of mine at North Carolina State University came back from a big conference, and he said this to me in the context of the Mountain Cloud Chemistry program where I was a part-time technician, that supercomputers were going to change the way weather forecasting on the TV even is going to be done. And WRAL, remember we're in North Carolina State University in Raleigh. WRAL was literally across the street from the campus. There was a period of time there where they said that they could give you a weather forecast down to your neighborhood. But they don't do that anymore because that big supercomputing modeling of atmospheric didn't turn out to be all that accurate than guys doing it by eye. National Center for Supercomputing Applications. They're in with Sandia National Laboratory. So what do? Is a hub for transdisciplinary research and digital scholarship. Collaborators from around the globe unite to address research grand challenges for the benefit of science. Yeah, but specifically, what are your current projects? Now, you can see how I look at things. Funding and proposal support. Compliance news. Featured stars. Project to study. University wins study of emerging pathogens. NIH grant to fund Alzheimer's. New website compiles ocean data from landmark science and 19th century science. But where's the supercomputing? Researchers propose new structures to harvest untapped fresh water. New study finds that reading for pleasure can strengthen memories in older adults. Oh, I'm an older adult, and I sit here in front of this, and I do this kind of bullshit to strengthen my mind. I don't have to go to the library. It's right here. Researchers propose new structures to harvest untapped sources of fresh water. So this guy Praveen Kumar, I've known somebody named Praveen Kumar, but I doubt it's the same guy, have proposed fresh water by developing infrastructure to harvest oceanic water vapor. Sounds very Israeli. Prairie Research Institute, Executive Director. Atmospheric Sciences. The Mountain Cloud Chemistry program was centered on the Forestry Department and the Atmospheric Sciences Department at NC State, with the Soil Sciences Department as an affiliate. Professor, National Center for Supercomputing. Professor, Center for Digital Agriculture. Remember, I wrote the code for the decision aid for post-emergence herbicide use, controlling weeds. Personal profile. Oh, this guy is very smart. He went to the Indian Institute of Technology in Bombay, 1987. And then that's where he got his Bachelor of Technology. And then he went to Iowa State, Corn Belt, PhD, University of Minnesota. Oh, yes, they grow corn in the northern prairies in Minnesota too. But he's interested in hydrology. So we've wandered around, but we still haven't gotten to find out more about this guy. There's his telephone number. To understand mechanisms of plant responses to both rising atmospheric carbon dioxide, people phrase their research objectives in terms of the current headline mandates. I knew a researcher at NC State that altered her proposal to include no-till agriculture. Why her work fits into no-till agriculture, which was the new mandate at the beginning of the 1990s by the USDA funding arm. So, to understand what is being said here, we say, now my head's in the way a little bit, to understand mechanisms of plant responses to rising atmospheric carbon dioxide concentration and tropospheric ozone, with particular reference to photosynthesis and relating changes at the molecular and biochemical level to observations of whole systems in the field, meaning the plants themselves. Productivity. Established the potential of mitigation of atmospheric change. You see how it's really a reiteration of all the political, climate change, disaster ahead of us. He's just saying it a different way. That's the whole aim, because that's where the funnel with the big money is. Go back and read what these website or the research proposal top lines said. Go back through the years and key it up to the headline stuff. There was a time when ozone was the really big thing. The ozone hole. Ozone protects us from UV, ultraviolet radiation. Skin cancer goes up with more UVB. That's the shorter wavelength. Established the potential for mitigation of atmospheric change through the development of herbaceous energy crops. What are herbaceous energy crops? carrots, soybeans, corn, beets, wheat, barley, herbaceous, not as opposed to trees. Ag, field ag. Advance the development of accessible mechanistic mathematical models. Advance the development of accessible, remember the person I worked for did crop modeling? That's on computers. Crop modeling. You grow the soybeans or whatever in the computer. You get the weather profile for the last 10 years of a locality. It's like we had somebody contact us literally from the Northern Prairie, and she said, when I ran your crop model for here with our weather, it didn't even come close to our actual yields. And then the researcher I knew said, send me the weather, and then she, here's the key words, reparameterized her model to reflect the weather trends, climate trends of where that person was. And that person smiled because the model suddenly began properly emulating their field research results by reparameterizing it, which is, by the way, the critics of the so-called climate crisis, the critics, the ones that were the original modelers, say, I don't agree with the way they have parameterized the models. They've parameterized them in such a way as it looks like we have no future, but I'm not sure that that's actually the realistic set of parameter values that should be used. And when you use the ones I think are more appropriate, we don't have a climate crisis anywhere near the scare tactics. Establish the potential of mitigation of atmospheric change to advance the development of accessible, meaning it doesn't take a supercomputer to run it. You can run it on your PCs. The PCs we have today are better than the supercomputers of 1985. And they can do it out in the field on the tractor. Advance the development of accessible, mechanistic, the crop models were based on a certain kind of mechanism having to do with interaction with atmospheric things, but I think he's talking about something else within the biochemical system of the photosynthetic apparatus of the soybean leaf cells. Mechanistic mathematical models relating to environmental effects on photosynthesis to plant productivity. See WIMOVAC. As soon as it says VAC, UNIVAC. To understand the limitations to C4 photosynthesis, there's C3 and C4, and the adaptation of the process to cooler climates. Department of Plant Biology, School of Integrative Biology. Research to find new carbon capture potential. Windows into the deep past. University of Illinois experts share perspectives on food production and climate change on PBS NewsHour. Told you the propaganda is really important. Teams find reliable predictor of plant species persistence and coexistence. Haven't you, like when I look at my lawn from year to year, even the birds keep changing. They're claiming they've seen trends that they can computer model. That's what, I'll bet you that's what they're saying. The key to species diversity may be their similarity. Making a campus green spot more interactive and even greener. Changes are underway at the Plant Biology Greenhouse and Conservatory. Anywhere near the campus with a green thumb, come and visit it. Like many ecological scientists, the University of Illinois Plant Biology O'Dwyer has spent his career searching for ways to measure and predict how specific plant communities will fare over time. Now think about what I said about the crop modeler at NC State. In that case, it was soybean and maize. And then when somebody out in the prairie said it doesn't seem to predict what our plant growth results have been, she reparameterized it so her models were more local in their nature. That doesn't mean there was anything wrong with them. It just showed that it wasn't universal. Be careful of anybody who claims total universality. She was pretty damn smart. Which species in diverse population will persist and coexist? Which will decline? Which factors might contribute to continuing biodiversity? Whether pairs or groups of plants are likely to coexist over time. Using data from published studies, see this is computing, their approach reliably predicts the complementarity life histories of pairs of plants that while competing for many of the same resources, manage to thrive in a shared habitat. Ta-da! Let's get back to him. Hang on. This is going on forever. We don't want that. Where's the ripe? Where's the ripe thing? Where's the name of the project? Here it is. Let's go back to that page. Realizing increased photosynthetic efficiency. Right. Let's go to their homepage. This will be the end. RIPE. We went into the publications. Let's see what they have produced. June 28th, 2023. Is chloroplast size optimal for photosynthetic efficiency? Now, Dr. Long's name is last. The first two names, the first name is Polish, the second name is the Dutch guy. This stuff is all very international. June 16, 2023. Impact, remember they're looking at soybeans. Impact of pod and seed photosynthesis on seed filling and canopy carbon gain in soybean. The first author is Chinese. The second author is German extraction. The third is English extraction. The fourth is Chinese. The next one is probably German, and the last one is German, Dutch, or Belgium. And it was published in Plant Physiology, major journal. The one above was New Phytologist, that's a major journal. Seasonal climate conditions impact the effectiveness of improving photosynthesis to increase soybean yields. The first person is Chinese, the second one, we don't know, field crop research. So these are practical. Calvin cycle. What's Calvin cycle? It's got something to do with, I'm trying to remember. Does that got something to do with mitochondria? Calvin cycle in, it is photosynthesis. Give me an F. Some of this is refreshing me. The Calvin cycle, light independent reactions, biosynthetic phase, dark reactions, or photosynthetic. Okay. Photosynthesis is a series of chemical reactions that convert carbon dioxide and hydrogen carrier compounds into glucose. So the chloroplasts capture the rubisco and stuff, captures the light with the pigment help and converts it into something that allows the catalytic conversion of carbon dioxide into glucose, which is a long-chain sugar. It gives you diabetes. It floats around in your bloodstream and you have to have it, otherwise you wouldn't be able to move. The Calvin cycle is present in all photosynthetic eukaryotes, not necessarily in bacteria, fungi, and algae, and also many photosynthetic bacteria. In plants, these reactions occur in the stroma. That's in the peripheral area. See, this stuff up here in the thylakoids has to do with the actual light capture in the electron photosystem 1 and photosystem 2. In the fluid-filled region of the chloroplast outside the thylakoid membrane, these reactions produce products like ATP and NADP, TPH, and perform further chemical. It's a chemical factory. After the green part actually converts light into excited electrons that can be used to power specific chemical reactions. And then there's the regular enzymatic steps to do the finishing of the baking. I'm just interpreting what the hell these people are talking about. Evaluation of the effects of elevated CO2 concentrations on the growth of cassava storage roots by destructive harvest and ground-penetrating radar scanning approaches. Increased bundle sheath leakiness of CO2 during photosynthetic induction shows a lack of coordination between the C4 and C3 cycles. And they're showing corn, maize. High throughput analysis of non-photochemical quenching in, can you say some of those words? In crops using pulse amplitude modulated chlorophyll fluorometry. Do you understand what that said? Do you understand what that said? That's something similar to what I did in the Mountain Cloud chemistry program as a side project for myself in the late 1980s. High throughput analysis. So they're able to do 1 million readings of non-photochemical quenching. What's quenching? The light comes in and you want it to be captured by the thylakoid membranes in the chloroplast for maximum conversion to glucose. But there may be other things in the plant leaf that spoil the light capture. Non-photochemical quenching. Non-photochemical chlorophyll fluorescence quenching is a process in which excess absorbed light energy is dissipated into heat. I've never looked for that. This is the first time I've tried to do that search for the definition. Compare what this process takes place in photosynthetic membranes of plants, algae, and cyanobacteria. Those are the ones that have systems similar. I knew a gal who did her PhD thesis on cyanobacteria photosynthesis. This website uses cookies Yeah, but that, allow all. I want to see the picture. Where's the picture? Oh, no, this is the article. Non-photochemical quenching, from light perception to photoprotective gene expression. Henan University, China. I just want to get the picture. Never mind. Never mind. So I did the definition and I wasn't far off. Soybeans photosynthesis and crop yields are improved by accelerating recovery from photoprotection. Guard cell targeted overexpression of Arabidopsis. Have you ever worked with Arabidopsis? Do you know what it looks like? Hexokinase. What would be a hexokinase? Would that be some kind of sugar converter? Hexokinase 1 can improve water use efficiency in field-grown tobacco plants. What's hexokinase 1? Has it got something to do with some kind of non-digestible food, sugar? Come on. Go to the website. National level. See, I like to do this. Hexokinases phosphorylate glucose. Hexokinases phosphorylate glucose. What's the output of photosynthesis? Glucose. And then there are other steps to process it into things that the plant needs, or for transport, or for metabolism. Hexokinases phosphorylate add phosphorus to glucose to produce glucose 6-phosphate, the first step in most glucose metabolism pathway. This gene encodes a ubiquitous form of hexokinase which localizes in the outer membrane of the mitochondria. They're talking about what we have and plants have. It must be an essential step in all of life systems. Mutations in this gene have been associated with hemolytic anemia. See, this is talking about affecting humans. But we were talking about mitochondria. But basically, the biochemistry at a deep level is pretty much the same. Wonder why? Because we're all dependent on the sun one way or another. Almost all of us. All right, folks. Now, here's the deal. This is the RIPE Institute. I thought this was going to be supercomputing. To me, this looks like standard plant breeding stuff. To me, it looks like standard plant breeding stuff. But that's not what I thought it was going to be from when reading the introduction. But it does fit within it. There is limited understanding of the carbon assimilation capacity of non-foliar green tissues, like the stem, and its impact on yield and seed quality, since most photosynthetic research focuses on leaf photosynthesis. See, I can interpret what he's talking about. So it all started with a BBC article. We got into some research that this guy... Now, the deal is, if you read further along in the article... Now, notice the things, the publications of that research institute. I said it all sounds like standard plant breeding. And by the way, standard plant breeding does use Fortran programs in order to evaluate yields and stuff like that, plant selection and so on. The efficiency of photosynthesis in crop plants is well below the theoretical maximum, but has been hard to influence thanks to complex nature of the process. There are only thousands of genes. There are more than 100 steps coded for by even more genes. I just said that, giving millions of potential permutations. Which allele combination overall would be the hypothetical maximum in heaven? Did you understand what the use of the word? All right, you say mutation. Add a gene, there's a sequence, and between different individuals, humans, animals, plants, algae, there may be point differences, sometimes segment differences. The genes may be all of those different ones which perform the same function, may be all perfectly okay, but maybe slightly different efficiencies or something like that. Those are known as alleles. Different versions of the same gene at the same locus are called alleles. They're not called mutations. Mutation means something else. Here's the computing. For those millions of options, the software can identify those that will make the biggest improvements. Oh, wait a minute. Professor Long and his team have used powerful computers, Supercomputing Center, to build a digital twin simulation of the photosynthetic process. Just like the person I worked with did crop modeling, they have built a computer model of the photosynthetic process, presumably segmenting it into what they believe are different genetically controlled elements. It can tweak the process in millions of ways. Of course, they could just do a random. From those millions of options the software can identify, that is, they pretend to model what the total end result would be. See, there's 100 steps. Step A, step AA, step AB, step AC, and within AA there's AA1, AA2, AA3, which are gene alleles. From those millions of options, the software can identify those that will make the biggest improvements. Presumably they have identified actual alleles and have some way of measuring their actual in situ, in the plant, actual efficiencies. We then engineered these into plants, which means they're doing gene substitution. And if that result is an improvement in the glass house, greenhouse, first they plant them in the sand benches. So they're looking at individual plants, 3 or 4 or 5 plants in an experiment. Some will obviously not be producing anything out of the ordinary. We're not interested. But this is plant breeding, this is plant selection. Corn breeders grow a crop in winter in Florida, but something like soybeans, for the way they're doing it, they may be able to do things in the sand bench in the winter. And then they have the selections of envelopes of seeds they want to plant out in the field in the summer for the real trial. We then engineered these into crops, and if that result is an improvement in the glass house, then we take it to our experimental farm and test it in a real-world environment. But he doesn't hint at all how they do the recombination at a genetic level. That's already had promising results. Changes to the mechanism of photosynthesis in soybean plants has resulted in yield improvements of more than 20% in controlled environments, with field trials now underway. One of the things that they learned in corn breeding, all right, let me explain it this way, just so you understand something early on. Prior to the 1930s, when there was no hybrid corn available, people were using what were known as open pollinated populations. But they had been selected in such a way as that the crop that they were producing was fairly uniform. So let's say you grew an acre of corn using that, and let's say the acre produced 200 pounds, produced, let's say, 10 bushels of corn. But then certain companies like Pioneer Hybrid started to develop how they could produce hybrid corn, B73, Mo 17 only. Those didn't exist yet, but the predecessors, two really different maize populations, combined them in an efficient production way to produce hybrid F1 seed. So when they went out to the farm demonstrations and they got farmers to plow in a bushel of this beside 1 acre of this while you do your regular 99, we'll pay you. And then all the farmers come around at the end of the season at the harvest. And like when I took the experimental design course, graduate level course, Dr. Gary Krause, I can still picture him saying it. He said it was no problem. You didn't need any statistics in those days. Because when they put harvested a row of regular open pollinated corn and they put the corn on the ground in a pile, and then they harvested the row of the hybrid corn, which they had never seen, and it was like the pile was 3-4 times larger. You didn't need any statistics to convince the farmers to try F1 seed, hybrid seed. That was how big the difference was. But now, speaking in the 1980s, if a plant breeder working for companies developing new lines, better lines, if he can make a 1% a year increase in yield, he's got to use field trials that have the most different kinds of crop growing conditions and strong statistical methods, so he doesn't get fooled into offering a line that actually is no better than anything that's out there when he's saying this will give you more yield. Because they're struggling to get just a 1% a year increase in yield, however you do it. Better photosynthesis, less lodging. Lodging is the plant falls over on the ground and you can't harvest it. Insect resistance, drought tolerance, poor soil poison acidity, etc., etc., etc. It's also complex. So they're claiming that they're taking a new approach and they're going to do it at the genetic level under the guise of supercomputing modeling. We shall see. One last story. When Marcus Zuber was sending out, he's a USDA ARS plant breeder, particular year, he had some candidates, inbred lines, to be crossed with the Iowa stiff stalk synthetic, and he sent them out to various cooperative researchers in the central part of the United States and the south of the United States, for those people to grow them up and drew crosses and then evaluate the progeny. And he said that with respect to Mo 17, the inbred line that he developed, that year he only had one envelope left after he sent all the samples out, 1 or 2 envelopes left. Now, that means that those people had to produce F1 seed in hybrid seed in Florida in the winter, and then take those to field trial, growing them in the field in Iowa, Minnesota, whatever, Illinois, during the summer. And he said in the fall, his telephone wouldn't stop ringing. They all wanted Mo 17. Because it combined not only with B73, the stiff stalk synthetic, but others, incredibly. And so he crossed his fingers and they went to Florida, and with the packet of seed he had, they grew out enough seed to be able to send the second-year samples to people to make sure that they weren't being fooled. And then it took off after that. It became the dominant cross of the entire corn belt. Mo 17 in combination with B73 and others like it. And if you were to look at a B73 plant, you couldn't believe what the F1 progeny looked like. Anyway, they're trying to take a new approach. They're at a supercomputing center. They're at a sustainability institute. They're at the University of Illinois Urbana, which is a big ag research university since forever. And we'll see how it turns out. And I saw it in a BBC article. That's what this is, right? A BBC article. I had to go to UK to find him. Steve A. Foriel, just an idiot running his mouth, saying see you in 73.